All right, welcome everyone. So this is our first video of the term. We're going to be getting into chapter 1, the biology of life, so exploring life. So in this first chapter, we're going to cover 3 basic parts. We're going to start out by looking at the common themes in life, so what unites living things together, as well as a brief introduction into evolution, so how we're all connected, and that explains the diversity of life. And then finally, we're going to end the chapter with how we study life, so talking about the scientific method. So let's start out with an easy question. What is biology? So biology is the study of life. So we are going to be studying living things. We're going to be starting out the term with chapter 1 with a brief overview of biology. And then we're also covering chapter 2 this week, which gets into the chemistry of life. And then we're going to build from there. Okay, so we're going to get our foundation set this week, and then we're going to build on it throughout the term. So in this first video that we're going to watch, we're going to learn about the hierarchical organization of life. So how living things can be organized. So I like these Amoeba Sisters videos. This is a great study tip, by the way. If you're ever confused about a biology concept, if you go to YouTube and you search Amoeba Sisters have put together quite a few good videos that help explain biological concepts in a fun way. So I highly suggest you use this as a resource if needed throughout the term, and you'll see me use these videos throughout the term as well. So we're going to start out with this one on hierarchical organization.
If I asked you to describe the biological levels of organization, could you do it? What would those levels be? Actually, what would be the point about caring about them in the first place? Well, biological systems are composed of many levels, and we care about them because when these levels are analyzed, it's possible to relate the levels not only to each other, but the whole system. All of our videos tend to focus on one or more of these levels, which is why we have chosen to make this video come first in our biology playlist. The first level that we are going to start with is the most basic living level that makes up all of life. It is the cell. Now, can something be smaller than a cell? Sure. For the cells that contain organelles, the organelles are obviously smaller than the cell they are contained in. Biomolecules are smaller than cells. We learn about biomolecules as they are major non-living components within cells. Atoms are smaller than a cell. You can even get smaller than atoms when considering subatomic particles. But for this video, we are going to start at the cellular level, a basic living unit. It doesn't matter whether you're talking about bacteria, archaea, fungi, protists, plants, or animals. If you're talking about something alive, you're talking about something made up of one or more cells. Now, when you have a lot of cells that are working together for a combined function, you end up with the next level, tissues. An example of tissue includes cardiac muscle tissue, which would be made up of many cardiac muscle cells. Tissues make up organs, which is our third level. You have a lot of organs in your body. An example of an organ would include your heart. When you have multiple organs working together, you end up with the next level, an organ system. Consider the digestive system. It includes your stomach, the small intestine, the large intestine, liver, etc. These particular organs work together to help you digest your food and to use the food for energy that you need for all kinds of cell processes. The digestive system is just one example of an organ system. But tie in all of the organ systems and what do you get? An individual, an organism, like you, or this platypus. He's got organ systems too. Now, if you have a lot of platypus together, what's the plural of platypus? According to dictionary.com, you can say platypi, which sounds especially fancy. And you have a population. This population of platypi can breed with each other. And they're all the same species. It's a platypi population. Great YouTube channel name right there. But include other populations such as fish, algae, and plants along with the platypi, and now you have a community. A community includes many populations, but it does not include abiotic factors. Abiotic factors are factors that are non-living, like rocks. Abiotic factors are not considered in the community level, just biotic factors. Biotic factors, on the other hand, are living. When abiotic factors are considered, such as the rocks, the temperature of this environment, the water, and the biotic factors, such as all the organisms we mentioned before, we now have an ecosystem. Well, it turns out that many ecosystems can be found in different regions on the globe. If talking about this collectively, you are at the level of biome. An example is a desert biome or a rainforest biome. You will find desert ecosystems or rainforest ecosystems in multiple places on the globe. But when talking about a desert biome or a rainforest biome, you are considering all of these ecosystems. Now, what happens when you put all the biomes together? Well, now you are talking about the level of the biosphere. The biosphere includes anywhere on our planet where life is supported. It is the sum of all of the biomes, all the areas where organisms live. Well, we're finished with our tour of the biological levels of organization, but remember, this is just an overview. The more biology you learn, the more you will discover how these levels relate to each other and the whole system. That's it for the Amoeba Sisters, and we remind you to stay curious.
So that was our first video that goes through the hierarchical organization of life. Now notice it started with cell as the lowest level, but in this course we're going to go even smaller, okay? So if we take a look at our complexity of life, we can organize it into a lot of different scales, okay? So from the video, it started at the cellular level, and so we can see it at the cellular level with, for example, they showed like a muscle cell, and then from there we get bigger and bigger and bigger, more inclusive, or we can get smaller and smaller, okay? So we're gonna do both in this video, okay? So we're going to go through each one of these levels and describe them, okay? So we're gonna start out with the cellular level. If we have, multiple cells together, we get into this tissue, so we'll talk about that. Multiple types of tissues form the organ. Multiple organs form an organ system, and then finally we get the organism. If we have multiple organisms that can interbreed together in a same location, we get a population. And then from there we can get different species interacting together to form a community. So that's the biotic life. And then if we add in the abiotic factors such as the land and the climate and the topography, we get an ecosystem. And then if we put all the living things together, all the ecosystems together, we get our biosphere. Okay, so that's becoming from most exclusive to most inclusive. And then we can get even smaller than a cell. Okay, so from the smaller the cell, we can get to the molecular level. So inside our cells, we have things called organelles. So those are like the organs of a cell. And then from there, the organelles are made of molecules, and molecules are made of atoms. So we're going to talk about these, but just realize when we go through the hierarchical organization of life, note that not all organisms have all of these levels. So for example, a single-celled organism, the organism level, since it's single-celled, it's only one cell, it will not have an organ system, an organ level, or a tissue level because it's simply one cell. So just be aware of that, okay? So let's get into the complexity of life. So let's start out by looking at the basically the organism. We're going to, here you can just see basically starting with our fish. Okay, so an individual organism, and then if we have our individual organism swimming with individuals of its same species, that forms the population level of life. And then from there, we have the fish interacting with other living things, such as the mangroves and the seabirds and the seagrasses and other things. That forms the community level of life. And then if we include all of the abiotic factors, such as the precipitation, the climate, the topography, all of that, we get the ecosystem level. And then from there, if we put all of the ecosystems together, we form the biosphere level. So the biosphere is the most inclusive. Okay, so you can see this is the most inclusive, whereas in this particular slide, the population would be the level that is the most exclusive. But we can even get smaller. Okay, so starting with our little snapper here, we have our organism. So this is a single organism. And inside that organism, we have organs and organ systems. So you can see here, we have a nervous system, for example. So these nervous systems and muscular systems and endocrine systems and integumentary systems, they make up the organism. So we can look at this organism at the organ system level, looking at the different systems. Or we can look at the individual organs that make up an organ system, such as the nervous system is made up of the brain as well as the spinal cord and the different nerves throughout the body. Okay, so we could study just the nervous system and the organs associated with it in this particular organism at the organ level. Or we can even go further and just look at the tissue that makes up a particular organ. Okay, so for example, nervous tissue is made of neurons. Okay, so, or that's a type of cell, but that's a type of nervous tissue. And so this nervous tissue, we can examine it and see how it works. Okay, and this nervous tissue is made up of a single cell. So this is the single cell. The single cell, by the way, is the smallest unit of life. So the smallest unit of life is basically, this is what's considered alive. Anything smaller than that is not considered alive. So we have our cell here. This is a neuron. And you can see inside that neuron, we have a nucleus and we have the DNA. And, if you know anything about neurons, they have axons and dendrites, and they basically send signals. So they're involved with sending communication, electrical impulses. So inside this cell, we can get even further. We can look at the organelles. So again, organelles, think of organs of the cell. Okay, so inside the cell, in some cells, we have little compartments that have different functions, such as the nucleus. Its job is to house the DNA. Or we might have the mitochondrium, which is the powerhouse of the cell. Or we might have the endoplasmic reticulum, which modifies and makes the proteins and lipids and things like that. So those are the organelles that make up the cell. Okay, so that's a smaller unit inside the cell. And this organelle, for example, the nucleus is not considered alive. So the smallest unit of life is the cell itself. Now we can get even more exclusive. So inside the organelle, we can look at the molecules that make up the organelle. Okay, so for example, inside the nucleus is a macromolecule known as DNA. Okay, so DNA is a nucleic acid, and that nucleic acid is a macromolecule. So it's made up of many molecules. And if we can even get smaller than that and look at individual pieces of molecules, which we call atoms. So atoms are the smallest unit of matter. Okay? So this is the complexity of life. So we're looking at basically in this picture, the most inclusive to the most exclusive. All right, going down to the smallest unit of matter, the atom, and then the smallest unit of life, being the cell. Okay. So here's a checkpoint question for you. Let's see if we understand this. Which of these levels of biological organization includes all of the others in the list? Is it the cell, the molecule, the organ, or the tissue? Okay, so which of these levels includes all the others in the list? So basically another wording of this question is, which is the most inclusive? Is it the cell, the molecule, the organ, or the tissue? If you said organ, I would agree with you. So if you recall, the organ contains tissues, and the tissues contain cells, and the cells contain molecules. So the organ is the most inclusive because it contains all three of these. Right? All right, so now that we know about the complexity of life, we're going to get into looking at cells. And we'll get more in detail of this in further chapters, but this is just kind of an overview. So we can take every single cell in the entire world and divide it into two categories. Is it prokaryotic or eukaryotic? So prokaryotic, pro means before, karyote means like kernel, which could be like nucleus. Okay, so this is basically prokaryote just basically means like an earlier cell, a very simple cell. Eukaryote, U means true. Karyote means kernel or nucleus. So eukaryote means true nucleus. So in the next few slides, we're going to compare these two types of cells. Okay, and remember, every single cell in the entire world can either be classified as prokaryotic or eukaryotic. And we're going to divide them based on their cell size, if they have organelles, the type of genetic information they house, or where it is, I should say, and then some examples. So in your notes, it's always a good idea to make like a table. Okay, so have prokaryote and eukaryote as the two columns. And then you can have as a third column on the side, cell size, organelles, genetic information, and examples. And then that way it's easier for you to fill it in. So, cell size comparison, prokaryote versus eukaryote. Do prokaryotes have organelles? Do eukaryotes have organelles? Where is the genetic information housed in a prokaryote versus a eukaryote? And then what are the examples? So we're going to go through each one of these points. All right? So let's start out with cell size. At the cell size. So from this chart, we're going to take a look at the cells. First, let's get acquainted with this figure from your textbook. So if you notice on the left-hand side, it's this will be the biggest, and then the right hand will be the smallest. Okay, so you can see atoms are the smallest. Here's 10 meters, that's the biggest over here. So we're getting, size, we're getting larger as we go left. So prokaryotes, an example of prokaryote is bacteria. Okay, so here are our bacteria. And bacteria, they lie around 1 to 10 micrometers on average. Okay, so they're very, very small compared to our eukaryotic cells. So eukaryotic cells, for example, plants and animal cells, they range from about 10 to 100 micrometers. Now, if you think about it, you're going to learn more about these these units of measurement in this week's lab. Okay, so a micrometer is 1 times 10 to the negative 6. Okay, so that's 0.000001. Okay, so imagine a decimal point with 5 zeros and a 1. That's a micrometer, so very, very, very small. A nanometer is even smaller than that. It's 10 to the negative 9. So imagine, a decimal point with 8 zeros and a 1 after that. That's very, very, very small, okay? So you can imagine that most bacteria, they're really, really tiny. We cannot actually see them with a normal microscope. You can barely see them with a light microscope, if you can, okay? Most likely you can't. You have to view them with an electron microscope. Eukaryotic cells, such as plant cells, animal cells, fungus, protists, they can be seen with a light microscope, the type that you would use in a normal, lab if you had a lab on campus. Because they range from 10 to 100 micrometers in length. So you would be able to see, for example, if we took your cheek cell, swab of your cheek cell and put it under a microscope, we would be able to see it because you are made of eukaryotic cells, which are larger. So prokaryotic cells are very, very small compared to eukaryotic cells. And just note that eukaryotic cells, they have a great range in size. So for example, some of our cells, some cells you can see with the naked eye. For example, ever have an egg for breakfast? Okay, so that is one single cell, and you can definitely see that without a microscope. Okay, there are some nerve and muscle cells in the blue whale, the largest mammal on Earth, that are very, very long. Okay, so they can be the one in the blue whale can be, I think, 6 meters in length, so very, very long for those nerves. So we have some cells that can be very, very big, but most of our eukaryotic cells range from 10 to 100 micrometers, whereas our prokaryotic cells range from 1 to 10 micrometers. So think prokaryotic cells small, eukaryotic cells larger. Okay. Now here is another view of a prokaryotic cell versus a eukaryotic cell. Now first off, you should see right away one big difference, the size, right? Okay, so you can see the eukaryotic cell is definitely a lot bigger than the prokaryotic cell. Also, the eukaryotic cell is a lot more complex. So there are a number of different membrane-bound organelles inside the eukaryotic cell. Inside the prokaryotic cell, there are no membrane-bound organelles, okay? So for organelles, you say yes for the eukaryotic cell, no for the prokaryotic cell. Okay, so prokaryotes, they are simple in structure. Eukaryotes, they're more complex in structure. Prokaryotes, they lack most organelles, okay? Eukaryotes, they have many different types of organelles. You can see just from this figure, we have, a number of different types of organelles just shown in this picture here. Now, as far as genetic information goes, eukaryotes have their genetic information stored in a nucleus. The nucleus is a double membrane bound organelle, okay? So the DNA is housed inside this double membrane bound nucleus. So that's unique for the eukaryotic cell. The prokaryotic cell does not have a nucleus, okay? So the DNA is just what we call, floating around in the nucleoid region. Okay, so that's not, separated from the rest of the cytoplasm. Okay, so it's just hanging around in the nucleoid region of that prokaryotic cell. Okay, so just to review, our prokaryotic cell is small, eukaryotic cell large. Prokaryotic cell has no organelles, Eukaryotic cell has many. Prokaryotic cell has a nucleoid region where its DNA is housed. The eukaryotic cell has a nucleus, which is a double membrane structure where the DNA is housed. Okay, and then finally for examples. So I already told you that we can take all living cells and divide them into two categories, prokaryotes or eukaryotes. We can also arrange them into 3 domains. So we have 3 domains of life. That's our classification. So the prokaryotes, they actually have 2 of the domains. They have domain bacteria and they have domain archaea. So those are 2 of the 3 domains of life. The third is domain eukarya, which, hey, eukaryotic cells, that makes sense. Okay, so domain eukarya has the eukaryotic cells. Domain bacteria and archaea, they have the prokaryotic cells. And you can see just from the slide here, 2 micrometers. So this is less than 10. Okay, so these are definitely small, single-celled, very simple or creatures there. Okay, so bacteria is a domain that has made of prokaryotic cells. Archaea is a different domain. Bacteria is everywhere. It's on your skin, it's inside of you. Some can be symbiotic, some can be parasitic, can cause disease. Archaeans, they live typically in extreme environments, such as like the hot springs or high salinity environments, or high acidic, high basic environment, or very basic environments. So they can survive they're what we call extremophiles. Okay. Domain eukarya, those are again the organisms that have eukaryotic cells. And we typically divide them into 3 or 4 classifications. 3 of them are known kingdoms. So we have kingdom Plantae, so plants, the autotrophs, the ones that photosynthesize, that make their own food. Animals, kingdom Animalia, so those are the heterotrophs, the ones that consume their food through ingestion. And then kingdom Fungi, those are the chemiotrophs, so they're the ones that absorb their food. Actually, they're heterotrophs as well, but they absorb instead of ingest their food. They do not photosynthesize. And then we have one last classification. Notice this doesn't have kingdom in front of it. So Protists is just kind of the catch-all group. It's the group that we cannot classify as a fungus, a plant, or an animal. Okay, so we just put it in the Protist classification. So plants and animals, they are all multicellular. Animals are all multicellular. Plants, they have cell walls, they have chloroplasts. Animals, they lack cell walls. Fungus, they also have cell walls, but they're made of different type of cell wall. They can be unicellular, so think of yeast that you use to make bread or beer or wine. So that's a single-celled fungus. So those, can be, they can be multicellular or they can be unicellular. Protists, they just basically are not animals, not plants, or not fungi, but are made of eukaryotic cells. They can be single-celled, like these little paramecium and the amoeba there, or they can be multicellular. So think of like kelp, okay? Kelp, or the big sea algae. They look like plants, but they are not considered plants. So they are protists. So that's how we can classify some diversity of life. So we can divide them into every single organism into one of these 3 domains. And if it's found in domain eukarya, we can further classify it into one of these 4 groups. So checkpoint question, to which of the 3 domains of life do we belong? Of course, we belong to domain Eukarya because we are made of eukaryotic cells. And of course, we are kingdom Animalia because we eat, we ingest our food. Okay, so let's do a concept check, make sure we're understanding everything. So which of the following levels of life's hierarchy is not appropriate when referring to the two of life's domains, archaea and bacteria? The population level, the organism level, the organ level, or the molecular level. So which one of these levels can we not, refer to when we're talking about archaea and bacteria? If you chose the organ level, I would agree with you, because archaeans and bacteria, they don't have organs because they're single cells, right? So we can talk about them at the organism level or the molecular level, what makes them, DNA and stuff inside, or the population level, like a colony of bacteria. But we cannot talk about them at the organ level because they don't have organs, okay? They're single cells. So C is the correct answer. All right, so that concludes our complexity of life theme. So now we're going to get into evolution. So we're just going to briefly touch on this topic, but we will weave through this topic throughout the term. So evolution, when we're talking about all the living things on Earth, we can look at it and see similarities, okay? We already described that all living things basically can be just have two types of cells, either a prokaryotic cell or a eukaryotic cell. So it's distinguished by its unity, but it's also distinguished by its diversity. Okay, we have anywhere from 10 to 100 million different species. I think we have like 1.8 million that we've been able to describe, and there's anywhere from 10 to 100 million out there. So we have lots to discover. And so there's a lot of diversity out there. And so the simplest explanation for all of this unity and diversity is evolution. Okay, so what is evolution? So evolution is the process of change that has transformed life on Earth from its earliest form to the vast array of organisms living today. So we're going to talk about evolution in detail towards the end of the term, but for now we just need to kind of get a basic understanding of it, okay? So let's learn a little bit about that in regards to natural selection. So as I stated before, evolution, it's basically the core theme of biology. So we need to, understand the basics of evolution. And the theory of evolution by means of natural selection, the idea has been around for a while. Charles Darwin is thought of as the father of this idea, though he wasn't the only one. There were others that were involved in designing this or coming up with this theory. But he was the one that published on origin of species by means of natural selection. So that came out, I can't remember off the top of my head, I believe it was 1865, something like that. And that book had, took off and was a bestseller and really brought, some understanding to our world. Because prior to Darwin publishing that book, people just thought that everything was static, okay? So all living things were here and they've been here and they've never changed, okay? So a squirrel was always a squirrel, and there's no link between species, okay? They were put here on Earth and there's no connection between species. So he was the first to find that connection, that unity. So how do we do that? Well, he based his theory of evolution by means of natural selection based on observations, which is how we use the scientific method. And so he found that within populations, within a population of any species, there are heritable variations. So what does heritable mean? Okay, that means that it's heritable, think heredity, genetics, okay? It's inherited. So think of traits that you have that you got from your parents, okay? You inherited those traits from your parents, and they inherited those traits from their parents, and their parents, and their parents. Okay, so there is a genetic link. It's in your DNA. So within a population of anything, we can talk about anything. We can talk about squirrels. Within a population of squirrels, there are variations within that population. Okay, no squirrel is identical. Even though you might think they look alike, there are slight differences between individual squirrels within a population. And those differences, they have a genetic difference, okay? So it's in their genes. And so those genetic differences come from their parents and their parents' parents and so forth. Also, within a population, there is an overproduction of offspring, okay? So we tend to produce more offspring than the environment can support, okay? So within a population of squirrels, there will be more squirrels being born than there are resources to support those squirrels. So what does that lead to? That leads to competition, okay? The squirrels are going to be fighting for the limited resources. So that leads to what we call unequal reproductive success. So some squirrels will survive and others will die, okay? So the squirrels that survive, they are the ones that have the best genes. They're the ones that have the best genetic makeup for that particular environment at that particular time. They're the strongest. It's the survival of the fittest, right? Okay, you've heard that term. And that's what natural selection is all about. We have unequal reproductive success leads to evolution of adaptations in a population. So the ones that are surviving are reproducing and passing on those traits that allowed them to survive and reproduce. They're passing those on to their offspring. The ones that don't have the traits that, are good in that particular environment, they die off, and so do those traits, right? So over time, those traits will be passed on to the offspring, and those offspring will survive and pass them on to their offspring. And over time, the population as a whole will become better adapted to the environment, okay? So unequal reproductive success leads to evolution of adaptations in populations. So let's do an example. So let's say we have a population of beetles. And notice within this population of beetles, there is some variation in traits. And that variation is inherited. So you can see that variation is in relation to the color of their carapace or the exoskeleton. So some of them are darker, some of them are lighter in coloration. And they inherited that trait from their parents. So it's in their genes. Okay, so a darker colored beetle probably had darker color parent beetles. The lighter colored beetle probably had lighter color parent beetles. So they have lighter colored genes. The darker color beetle has darker colored genes. So within this particular environment, which one has the better adaptation? Well, the ones that are darker color, because they blend into their surroundings. They are camouflage. So in this particular environment, the lighter color ones are at a disadvantage. They are more vulnerable to predation. So they're more likely to get eaten before they reproduce. And if they're more likely to get eaten, then their light colored genes will die with them. That way the darker colored beetles, they'll survive longer and they'll reproduce and pass on their darker genes to the next generation. So if we go through several generations of this, over time, we'll see the population evolve to have more darker colored beetles. So look at the initial population, you see more lighter color. In the final population, you see more darker color. So evolution took place. It's just a change in the genetic frequency of a trait over time. That's it. Okay? That's what evolution is all about. So now we have this particular species of beetle is better adapted to this particular environment. Of course, the environment changes. Okay, so what might be the best adaptation now might not be the best adaptation in the future. Okay, so that's why, everything's always changing. Okay, nothing is going to be best forever. All right, so let's do a concept check to make sure we're understanding things. Darwin proposed the theory of natural selection. For the most part, natural selection acts as a filtering agent at the what level of life's hierarchy? Is it at the community level, the population level, the individual level, or the organ level? So natural selection acts as a filtering agent at what level? If you said population level, I would agree with you. Okay, it cannot act at the individual level, like some of you might have thought, because you do not change. Okay, you were born with a certain set of genes, and those genes, they don't change. Okay, you cannot suddenly go from being a light colored beetle to a dark colored beetle, right? Okay, that cannot happen. Your genes say light color, you will always produce light colored beetle genes. There's no changing. The population, however, can change over time. Okay, so populations can change over time, individuals cannot. So that's a misconception that some students have. So just remember, populations can evolve, individuals cannot. All right. So as far as evolution, we already know that how it works. Okay, so we know that natural selection is what promotes adaptation. Okay, and that leads to evolution. Now, when we think about evolution, we can use this as a tool to understand life's diversity. So when we look at all the species on Earth today, all 1.8 million that we've been able to describe, each species has a family history. So we can create what's called a family tree, or also known as a phylogeny. So a phylogeny is just basically a branching tree of life that extends back through time, where we can see where species may have had an ancestor in the past. And so we can see how connected two species are to each other and how species are related to others. So we're going to look at one of these phylogenies and be able to interpret it. So here is a phylogeny of some of our common mammals of today. So you can see in, these are the families of different mammals. So Procyonidae, those are the raccoons, Mastidae, the weasels, the otters, and so forth. So anything to the right here are existing species, or in this case, they're families of species. Okay, so in this particular example, as we move left, we're moving further back in time. Where we have a branch intersecting, that's called a node, and that indicates a common ancestor. Okay, so a period of time when we had a common ancestor. So from there, there was a common ancestor, and then the two families branched off from there. Okay. Here's another common ancestor. So here we have this branch forming with this group. Okay. And then this branch forming with this group. So we can see where the relationship is. So you can see that raccoons and weasels are more closely related to each other than they are to red pandas, because their common ancestor is more recent in time. Okay. red pandas are more closely related to weasels than they are to skunks because they share a more closely related ancestor than they do to skunks. If you wanted to know the ancestry of red pandas to giant pandas, they are related but distantly related. Okay, so you can see the red pandas are more closely related to raccoons and weasels than they are to the giant pandas. Okay, giant pandas are more related to like the other species, like bears and things like that. They're more of a bear. Okay, red panda, they did share a common ancestor a long time ago, but they're more they're more ancient in time. All right. So this is a way that we can see how things evolve through time and what is related to what and how closely related they are. So here's a checkpoint question. Explain the cause and effect of unequal reproductive success. So explain the cause and effect of unequal reproductive success. So first of all, what is the cause of unequal reproductive success? Do you remember from that slide? One is that we have variation within populations, and that variation is heritable. Okay, so heritable variation. And the other is overproduction of offspring. Okay, so those individuals with heritable traits best suited to the local environment produce the greatest number of offspring. So that means that we have unequal reproductive success. Okay, so unequal reproductive success is due to the fact that we have within populations variation, and the ones with the best traits survive, the ones with the not so good traits, they die off. Okay, so we end up with the survival of the fittest. All right, so the rest of the lecture, we're going to get into the last theme, which is the scientific method. So we're going to start out talking about what is science. So science is a way of knowing. Okay, so we are curious about the world and we want to understand it better. So we, with science, is a little different from, other ways of understanding things. It's an evidence-based process. So we have to use evidence to understand things. Okay, and that's, where conflicts arise, especially in, today's world with explaining things to your uncle or something like that. The scientific approach involves observations, hypotheses, predictions, tests of hypotheses via experiments, or additional observations, and the analysis of data. So that is what we call the scientific method. So we're going to talk about the scientific method in detail. And it's a very intuitive process. I mean, you've done the scientific method all the time and you don't even know you did it. Okay, so we always, notice something around us and we ask questions, and then we kind of think about it and come up with a guess, make predictions, and then we kind of conduct an experiment to test that hypothesis. And then we gather data, which either helps us support or reject our hypothesis. So that's a scientific method. And then finally, the scientific theory, that's like a hypothesis, but it's broad in scope. Okay, so hypotheses, by the way, are basically educated guesses that can be tested through experimentation. Theory is broad in scope and supported by a large body of evidence. So can you think of a scientific theory off the top of your head? Well, we already talked about one of them, okay, the theory of evolution by means of natural selection. So that scientific theory is broad in scope and it's supported by a large body of evidence. So we have a lot of evidence to support it. We have fossil evidence. We have molecular data, like DNA evidence to support it. We have all sorts of different evidence that is, varied and broad to support that scientific theory. And we can actually conduct it ourselves with artificial selection. So we can actually do evolution ourselves in like with plants and things like that. Other theories we're going to talk about, for example, cell theory later on. So that is a theory that has to do with cells. You may have heard of the theory of gravity or germ theory. So those are all scientific theories. So they're broad in scope and supported by a large body of evidence. So most likely those theories will never change. But in science, we never absolutely say fact because you never know. So we are evidence-based. So there could be a piece of evidence that comes along later on that might change parts of the theory, but not the whole thing, most likely. Okay, so we are always seeking out evidence to learn more about this natural world that we live in. So let's talk more about this scientific theory. So let's start out by observation. So what is an observation? That's just basically using your senses, right? So for example, let's say that you went to grab a flashlight and you flip the little switch on the side and it doesn't work. So that's an observation. Okay, the flashlight doesn't work. So the next step of the scientific method is you ask a question. Why doesn't the flashlight work? Okay. Next, you might come up with some hypotheses. Okay, so one of them, like my first hypothesis would be, I bet the batteries died. Okay, or another hypothesis could be like the bulb burned out. That's another possible answer to your question, right? So a hypothesis is just basically an answer to the question that can be tested. Next, what do we do after the hypothesis? We come up with a prediction. So prediction is predicting the results of your experiment. So for my first hypothesis, if the batteries are dead, well, I predict that if I replace the batteries, the flashlight will work. If I replace the bulb, because the bulb's burned out, the flashlight will work. Okay? After you predict, then you perform the experiment. Okay, so for the batteries, I replace the batteries, and then I flip the switch, and if it works, then my hypothesis is correct, or I accept it. If it doesn't work, then it's something else. If I replace the bulb and it works, then I know I support my hypothesis. If it doesn't work, then I reject my hypothesis. So we test through experimentation. Okay. In this case, the results were that the flashlight works when we replace the bulb. Okay, so in this case, we had the first hypothesis was not supported, but the second hypothesis was. All right. Now, again, we do this every day. We just don't actually stop and say, what's my hypothesis? What's my prediction? But, we do this every day intuitively. So we always kind of, think about the problem, come up with, a possible solution, and then test that solution, right? So that's just something that we do with science as well. So what is the main requirement for a scientific hypothesis? What do we need to do to create a good hypothesis? So for example, if we go back to our last experiment, why doesn't the flashlight work? What is a bad hypothesis for that? Okay, so that would be something that cannot be tested. Okay, why doesn't the flashlight work? Because I don't like it. I don't know. Because God made it not work. That's not testable, right? Doesn't mean you can't believe in God or whatever. I know I'm not talking about your religious beliefs. It's just we can't test it. So what is the main requirement for a scientific hypothesis? It must generate predictions that can be tested by experiments or gathering further observations. So your hypothesis has to be testable. Okay, that is the main point of a hypothesis. It is an educated guess that you need to be able to test, and that your hypothesis also needs to be falsifiable. By the way, you can't just say, for example, if I go back, my hypothesis can't be that, why doesn't the flashlight work? It could be the batteries, or it could be something else. Okay, that's not a good hypothesis because that's not falsifiable. Well, it could be the batteries, but it could be something else. Well, how do I prove that it's something else, or the batteries? How is that falsifiable? So it needs to be something solid. Okay, batteries are dead. So that is falsifiable. Okay, I could prove that is not true if, if I change the batteries and it doesn't work. So the scientific hypothesis has to be testable, and it has to be falsifiable. I have to either support it or reject it. No in between, no gray area. You don't want to partially support a hypothesis. All right. Now, when we conduct an experiment, we want to make sure that that experiment is going to test our hypothesis. So we want to make sure that we're only manipulating one component of the experiment and observe the effects of that change. So when we're manipulating just one factor, it's called the independent variable. So independent variable is the manipulated variable. It's the variable that we ourselves are changing in that experiment. The dependent variable is what we're measuring the outcome. That depends on the independent variable, for example. Okay? So for example, if we're doing a controlled experiment, we're going to have an independent variable, which we're manipulating, a dependent variable, which we're testing, and then, we will have a couple treatment groups. One might be the experimental group that will change, and then we might have a control group, which we keep the same. So for example, let's say that we do an experiment on plant growth with different fertilizers. Okay, so in my experiment, this will be a controlled experiment where I will use a couple different types of fertilizers on my plants and see which one helps them grow the best. Now, in this case, I'll have experimental groups. My experimental groups will be the plants that receive different types of fertilizer. So some plants might receive one type of fertilizer, some plants will receive another type. And then I'll also have a control group. The control group is the group that doesn't receive anything. It does not receive the treatment. So in that case, no fertilizer group. Okay? And we want to use a control group for comparison, because if we're only using plants that receive fertilizer and they all grow the same, we don't know if it's the fertilizer that caused them to grow or something else. So if we have a control group as comparison, and the control group, for example, does not grow very well, then we know it was the fertilizer that helped them grow. So we need a control group for comparison. So in my case, I'll have two groups that are experimental groups, one testing one type of fertilizer, the other testing the other type, and then a control group that receives no fertilizer. Now in this case, what would be my independent variable? What am I manipulating here? What is the treatment that my plants are receiving? They're receiving fertilizer. Okay, so my independent variable in here is the type of fertilizer. So one group gets one type, one group gets another type, and one group gets no type. And then what is my dependent variable? What am I measuring? In this case, I'm measuring their growth. Okay, so I'm going to check out how much they grow each week. Okay, so the dependent variable depends on the independent variable. Their growth rate depends on what type of fertilizer they got. All right, so this is how we set up our experiment. Now, hypotheses can be tested using controlled experiments. So we want to use a control group, an experimental group, to basically test just the effect of a single variable. We don't want to use multiple variables in our testing. So sometimes when I see students, like if we did this in an in-person lab, some students get overzealous and start using multiple variables, and that can get confusing because you don't know if it was the first variable that caused the change or the other variable. So we just want to stick with a single variable. So we're going to look at an example of a situation where we have, in this case, we have mice. And we're going to look at how researchers found that these mice, they didn't match their habitat, and they had higher predation rates than the camouflage model. So we're going to take a closer look at that. And so, for example, researchers found that mice models that did not match their habitat had higher predation rates than camouflage models. So basically, we're going to look at two types of mice, and one of them is more, darker color, one of them's lighter color. And the lighter color one lives in the beach environment, which is lighter in coloration. The other one lives in the inland environment, which is a darker color. So obviously we predict that the lighter color mice will do better in the lighter color environment because it's camouflaged. And if we put them in the inland population, then they're probably going to get eaten because they stick out. Okay, so we're going to take a look at this example. Hypothesis, they can be tested in humans. Okay, so this is not something that is just in animals. We can do this in clinical trials. So for example, when you get into the health science field, there are clinical trials that can be tested, where we have individuals maybe testing a drug trial. Sometimes it's a double-blind study where we don't know if the individual is getting the actual drug that is being tested or a placebo. So what's a placebo? It's, it looks like the drug, but it's just a sugar pill. And then, you can test side effects and things like that. As well as retrospective or prospective observational studies. So we can kind of test, what happens through observation as well. So here is our example. We have our two mice. They're the same species, but they have different morphs. So you have different populations. One lives in the beach area and one lives in the inland. So you can see that the inland population is more brown. It's got a white belly, but more brown. So it's more camouflaged in its environment. The beach population, it has less brown. It's more white. And so it blends in better with the sand on the beach. So let's see if we can interpret the data. In this experiment, both dark and light colored mouse models were placed in each of the two environments, beach and inland, and predation was measured on camouflaged and non-camouflaged mice. So in this experiment, what serves as the controls? So what would be the control group in this case? Would it be the light-colored mice in beach populations, the light-colored mice in inland populations, the dark-colored mice in inland populations? Both A and C are correct, or both B and C are correct? So which one of these is considered the control group? The group we're not changing. If you chose D, I would agree with you, okay? So remember the light color, that's their natural environment, so we're not changing anything. The dark colored mice in the inland environment, that's their natural environment. We're not changing anything. So these are the control groups. When we put the beach mouse in the inland environment and the inland mouse in the beach environment, we're changing things big time, right? Okay, so that's the experiment. But the control groups are just when they're in their natural environment. So D is the correct answer. All right, so now we got to interpret the data. So the results of the experiment are shown in the table below. For this experiment in the beach habitat, what are the non-camouflage models? So which are the non-camouflage models in the beach environment? Are they the dark colored mice on the beach, the dark colored mice in the inland habitats, the light colored mice on the beach, or the light colored mice in the inland habitats? So in this experiment, what are the non-camouflage models in the beach habitat? If you chose A, I would agree with you. So which ones are not camouflaged in the beach environment? That would be the dark color mice. Okay, so they stick out, right? So A is the correct answer. So according to the table, is the following hypothesis supported? Actually, this is more of a prediction, by the way, because it's an if-then format. Predictions tend to be if-then format. If camouflage coloration protects mice from predation, then mice with coloration that does not match their habitat will be preyed upon more heavily than the native mice in that habitat. So basically, if you're not camouflaged, you'll get eaten more often. So does the data support that? So let's look at the beach environment. We have 2 being attacked for the camouflaged ones, whereas 5 were attacked for the non-camouflaged. So that's a 71% attack on the non-camouflaged. Whereas in the inland, we had 5 getting attacked for the same colored, 16% for the different colored. It's a 76% change. So based on that information, was the hypothesis supported? If you said yes, I would agree with you. So we see that there is a strong correlation between the non-camouflaged models being attacked. Okay, so they stick out, right? The brown mouse in the white environment sticks out, so it's going to get eaten. And the data supports that, 76% chance. All right, so here's a question for you. So hypothesis can be tested using controlled experiment. Just checkpoint question. In some studies, researchers try to match factors such as sex, age, and general health for subjects in the control and experimental group. What is this experimental design trying to do? So what are we trying to do if we're trying to match factors such as sex, age, and general health for subjects in a control and experimental group? What is this experimental design trying to do? Well, what are we trying to do? We're trying to ensure that we only have one variable that we're testing, right? So if we're trying to match 2 factors such as sex, males and females, age, the 30 to 40-year-olds versus the 40, 50-year-olds, general health subjects, okay? So that we're trying to make sure that the groups differ in only one variable, okay? And that's going to create the best experimental design. So we want to make sure that we're controlling for all other factors if possible. We just want to have one variable. All right, so that's it for our first chapter. The next video, we're going to cover chapter 2, the chemistry of life.